Hello everyone, BiyokimyaVET channel is here with the topic titled Enzymes. Please visit biokimya.vet website for a more comprehensive document of the topic in PDF format. You can find the link in the descriptions. And please do not forget to subscribe, like the video and turn on notifications before starting. I wish it will be useful. Let's get started. And here you see the contents of this topic that I will start with the definition of enzymes and give some brief information about the history, and we will look structural enzymes and then terminology, locations, and we will finish with the coenzymes. And enzymes actually are a subject that can be talked about in depth for a long time, my friends, but we will not do that. Our main focus in this topic is to provide you with basic enzyme knowledge. And the first title is Definition and History. Most biochemical reactions are catalyzed by organic substances with a protein structure. These biological catalysts are called enzymes. Enzymes are defined as biocatalysts that allow biochemical reactions to occur rapidly under normal conditions and constitute a fundamental characteristic of living tissues. Enzymes are molecules that catalyze a chemical reaction without themselves being broken down or modified. They are reaction catalyzed biological systems, chemical agents that allow biochemical events to take place in the body in a manner compatible with life. Enzymes and the branch of biochemistry dealing with them are called enzymology. And the word enzyme was first used by Wilhelm Kuhne and means fermented in Greek. It is still an important topic of interest in biochemistry today. And William Friedrich Kuhne was a German physiologist. He was born in Hamburg and is best known today for inventing the word enzyme. Another branch dealing with enzymes is clinical enzymology. It is the branch of science that includes studies and tests related to the diagnosis and differential diagnosis of diseases, prognosis, monitoring of the treatment process, and monitoring of abnormal organ functions by measuring enzyme activity in serum, plasma, urine, or other body fluids. And here you see some list of enzymes we measure in clinical laboratory: the ALP, alkaline phosphatase, alanine aminotransferase, aspartate aminotransferase, CK, creatine kinase, glutamate dehydrogenase, and the others. And by measuring the enzymatic activity of these enzymes, we can assess the tissue functions or the tissue integrity. Structure of enzymes. Except for a small group of catalytic RNA molecules, enzymes are proteins. Enzymes may contain only proteins in their structure, or they may also contain a metal ion as cofactor, such as glutathione peroxidase contains selenium, carbonic anhydrase, zinc, catalase, and cytochrome, iron, and we can increase the examples easily. The protein structure of the enzyme determines the substance it will act on and the type of reaction it will catalyze. Enzyme terminology. Most enzymes need an additional activating substance to become active. The part of the enzyme that is made of protein is called the apoenzyme. The factor that activates the apoenzyme is called the coenzyme or cofactor or cofactor. In real, the coenzyme, we use the organic substances which are required for the enzyme activity. The cofactor refers the ions. A complete enzyme, a holoenzyme, is made up of the apoenzyme, coenzyme, and or cofactor. There are some definitions regarding enzymes we need to know. One is processing group. The cofactor part of the holoenzyme is one or more inorganic ions such as iron, magnesium, zinc for some enzymes, and an organic or metal-organic complex molecule called coenzyme for others. If a cofactor forms a complex with the apoenzyme that is not easily cleaved, then this cofactor is called a processing group. And the example is porphyrin moiety in peroxidase enzymes, proenzyme or proenzyme or zymogen. And some enzymes are synthesized as an inactive protein or inactive enzyme precursors. They are called proenzymes or zymogens. And the example is the precursor of chymotrypsin is the chymotrypsinogen, or trypsinogen is the precursor or the zymogen of trypsin. Kinases. Sometimes another enzyme is needed for an enzyme to change from its zymogen form to the active enzyme state, and such activating enzymes are called kinases. And a good example is the enterokinase, which converts inactive trypsinogen into the active trypsin. Isozymes or isoenzymes. Some enzymes have multimolecular variants that catalyze the same reactions in the same organism, and such enzymes that catalyze the same reaction but show molecular differences or the structural differences are called isozymes or isoenzymes. And commonly, I prefer to say isoenzymes, and a good example is AL. Substrate. It is the substance on which the enzyme acts specifically is called substrate. The substance formed from the substrate after an enzymatic reaction is called the product. Exoenzyme and endoenzyme. Enzymes that are made in a cell and then released into the extracellular environment where they will function are called exoenzymes. While enzymes that remain in the cell where they are produced, that is, enzymes do not release outside the cell and maintain their catalytic activity inside the cell are called endoenzymes. What are the locations of enzymes? And I mean here, in the previous slide, I defined the endo and exoenzymes. And here the main focus will be the cellular location of enzymes. And we know enzymes are synthesized inside the cell, and the vast majority are used for intracellular purposes. So thus they are endoenzymes. But for the digestive system enzymes, such as pepsin, chymotrypsinogen, or trypsin, they are released from the cell where they are synthesized, and they have the activity in outside the cell. And the other exoenzymes. What are the intracellular location of enzymes? It varies. For example, in mitochondria, there are enzymes such as TCA cycle enzymes or beta oxidation enzymes. In the nucleus, pyrophosphorylase, aerosines, carbon anhydrase, and cytoplasm glycolytic enzymes. But these are just some examples. There are numerous enzymes in different parts of the cells. And what about the blood? Blood also carry enzymes, but in two classes: functional plasma enzymes and non-functional plasma enzymes. These are the two classes of enzymes found in blood plasma. What about their concentration? Functional plasma enzymes in higher concentration in comparison to tissues, but for the non-functional ones, normally they are present in plasma but in very low concentration in comparison to tissues. For the functional ones, they have known functions. This means their substrates are always present in the blood. For the non-functional, no known functions because their substrates are absent from the blood. The site of synthesis for functional in liver, but for the non-functional plasma enzymes, different tissues. So they come from different sources, including liver, heart, brain, skeletal muscles. What about the effect on diseases, or the effect of diseases on the activity or the concentration of these enzymes? The functional ones generally decrease in liver diseases. For the non-functional ones, they increase due to the tissue injuries. And some examples here, such as clotting factors. And for the non-functional ones, we have many commonly measured in clinical laboratory. And for the non-functional ones, also we call them cellular leakage enzymes, because the main factor that causes increase their activity in blood is the cellular injury that will lead to the leakage of these enzymes into extracellular fluid and plasma. Enzymes are highly specific substances. However, specificity can vary. Many enzymes act only on a single substrate. They do not even act on a stereoisomer of a substrate. For example, lactate dehydrogenase acts only on L-lactate, and the amino oxidase only on the D form of amino acids. Sometimes enzymes show group specificity. For example, hexokinase, phosphorylase, all hexoses, which are those galactose, glucose, and mannose. Some enzymes are strictly specific for a substrate. For example, glucokinase phosphorylase only glucose, not the fructose. Another example is galactokinase, which phosphorylase only galactose, not the glucose. And some enzymes act on all substances of a certain class of compounds, such as pepsin acts on all proteins. The function of an enzyme as a catalyst is to increase the rate of a reaction by lowering the activation energy. The distinguishing feature of an enzyme catalyzed reaction is that it occurs when the confines of a pocket on the enzyme called the active site. The active site is a special region on the enzyme molecule that has the ability to bind substrate. It binds the substrate and the enzyme substrate complex is formed, and the product is released when the enzyme dissociates from the enzyme product complex formed by the conversion of substrate to product. Enzyme activity is controlled in several important ways to ensure that it is present at the right time, in the right place, and at the right concentration. There are some factors which they have effect on the control of enzyme catalytic activity, and the one is the induction of gene expression, such as steroid and thyroid hormones are the hormones that have effect on to increase the enzyme synthesis, and the synthesized enzymes can then be activated by polypeptide hormones. So induction of gene expression tells us there are factors inducing both synthesis or enzyme activity. And to give an example, steroid hormone, they induce the synthesis of enzymes, and the polypeptide hormones induce the activation of inactive enzymes. Non-covalent enzyme substrate interactions, another factor. And substrates bind to enzymes through multiple non-covalent interactions between binding residues in the active sites and complementary chemical groups on the substrate. And there are two models explaining non-covalent enzyme substrate interactions, and the one is Emil Fischer's lock and key model. Assume that the substrate and the active site of the enzyme are predetermined to fit together, such as the log and another is Daniel Koshland's induced fit model. And in this model, the active site is flexible. In the presence of substrate, a change in the tertiary structure of the protein causes the enzyme to undergo a conformational change to bind to substrate in the most correct form suitable for catalysis. And here you see the illustration of both models, lock and key model and the induced fit model. And it is easy to understand here in the lock and key model, there is a template here which is completely complementary with the substrate. In other words, we can say there is a ready-to-use active site in the lock and key model. But in the induced fit model, it is different. As you see, it is not complementary with the substrate. So substrates bind to enzymes through multiple non-covalent interactions between binding residues in the active site and complementary chemical groups on the substrate. This is especially true for enzymes with more than one subunit, such as multimeric enzymes. Binding of the substrate to one active site can sometimes increase the binding affinity at other sites. In the template model, as I mean in the lock and key model, the active sites have a rigid structure, and in the induced fit model, they are more flexible. And in the induced fit model, which may be more physiological, substrate binding to an active site causes a conformational change in the enzyme. And look at the illustration and compare the initial state and the state which enzyme substrate complex form, and resulting in a complete fit between the enzyme and substrate at all active. We can say there is a kind of analogy to a sock taking the shape of a food in the induced fit model. We continue other factors which they have effect on enzyme activity or the catalytic activity. The feedback inhibition. Allosteric interactions also play a role in feedback control of enzyme activity. In biosynthetic pathways, groups of enzymes often work together sequentially to carry out metabolic processes. The reaction product of the first reaction quickly becomes the substrate of the second, and so on. If the regulatory enzyme is the first enzyme in the pathway, it can be controlled by the end product. And when the end product rises above steady state concentrations, it can bind to an allosteric site on the regulatory enzyme, thus disrupting its active sites and inhibiting its activity. Reversible covalent modification. The catalytic activity of an enzyme can sometimes be controlled by covalent modification. An example of this is glycogen phosphorylase, which exists in either an active phosphorylated form or inactive dephosphorylated form. This enzyme has two polypeptide chain subunits, each with a serine residue that is phosphorylated at the hydroxyl group kinase or dephosphorylated by a phosphatase. Similarly, glycogen synthase undergoes reversible covalent modification. It is inactive when phosphorylated but becomes active upon dephosphorylation. Control proteins. Enzymatic activity can also be controlled by the binding of specific stimulatory or inhibitory proteins. The GS and GI proteins in plasma proteins are examples, and these proteins are so named because their combination with GTP activates them. And upon binding of ligands such as some polypeptide hormones to external plasma membrane receptors. These proteins can activate or inhibit membrane-bound adenylate cyclase and thus regulate intracellular cyclic adenosine monophosphate formation. And another example is the intracellular protein called calmodulin, which is activated after binding 4 moles of ionized calcium. And this activate protein then binds many intracellular enzymes and alters their activity. And the last one is proteolytic activation. In contrast to the above modifications, which are all reversible, enzymes can also be activated irreversibly, a process that will naturally occur only once in an enzyme's lifetime. These enzymes are synthesized as inactive precursors, zymogens or proenzymes, and are then activated by cleavage of one or more specific peptide bonds. Proteolytic enzymes associated with digestion in the gastrointestinal lumen are good examples for this kind of activation. Classification of enzymes. And the first class is oxidoreductases, or shortly redox enzymes. Enzymes involved in biological oxidation. And there are just two subclasses. The one is the dehydrogenases or reductases, and another one is the oxidases. And the dehydrogenases, they get the name dehydrogenase, which means removal of hydrogen. So they remove hydrogen. Actually, they remove proton, thus an electron, from the substrate in the presence of a suitable hydrogen acceptor. And these are the coenzymes, such as NAD, FAD. Transferases catalyze the transfer of specific groups from one compound to another, and such as the transfer of functional group, especially the functional groups such as amino, acid, phosphate, etc., from a donor to an acceptor molecule. And there are many subclasses such as transferases of 1-carbon units, transferases of aldehyde or ketone groups, transferases of acid groups, etc. The third group is hydrolases. They catalyze the hydrolytic reactions. Add water to substrate, causing hydrolases, the breakdown of covalent bonds by adding water. There are many examples, or numerous hydrolases enzymes or hydrolytic enzymes, such as lipases, amylases, all are hydrolases. Esterases, all cholinesterases such as nucleases, they are all hydrolytic enzymes. Liases, enzymes that cleave groups from the substrate, reacting or conversely adding bonds. In liases, there is a non-hydrolytic cleavage of carbon-carbon, carbon-sulfur, and some carbon-nitrogen bonds by addition of a group to the double bond. And we have subclasses such as the carboxylases, and carbonic anhydrase is a class of liase, aspartate amino liase, cysteine desulfitase, and the others. Isomerases catalyze intramolecular reorganization or conversion of one molecule to its isomer, and the conversion of isomers into each other by transferring a group from one position to another within the same molecule. For example, ketoses into aldoses, converting a fructose into glucose. There are subclasses, racemases, epimerases, cis-trans isomerases, etc. And ligases form carbon-oxygen, carbon-sulfur, carbon-nitrogen, or carbon-carbon bonds. And formation of these bonds by joining two molecules together in reactions that require energy and usually provided by ATP or other hydrolyzers of nucleotide triphosphates. So these formations are catalyzed by the ligases. These enzymes play important role in vital processes such as the repair and the synthesis of DNA and RNA molecules. And here are some examples. DNA ligase used to join single-strand breaks in DNA molecules. It plays an important role in DNA replication and repair. Another example is RNA ligase. Ligates breaks in RNA molecules. It functions in processes such as RNA splicing and RNA repair. Another example, amino acid tRNA ligase enables the addition of amino acids to RNA to tRNA molecules, and this is a fundamental step in protein synthesis. Enzyme kinetics and activity. Enzyme kinetics expresses the relationship between changes in experimental parameters and the rates of enzymatic reactions. The rate of an enzymatic reaction is expressed in terms of the amount of product formed or substrate converted to product per unit of time by enzyme action. The number of substrate molecules converted to product per unit time by a single enzyme molecule at optimal pH. At temperature 25 degrees Celsius and saturating substrate concentration is called the enzyme conversion number and is denoted by the symbol kilocalorie. The rate of an enzymatic reaction is related to the activity of the enzyme. The activity of an enzyme is the expression of the rate of the enzymatic reaction catalyzed by that enzyme by the amount of substrate converted to product at a certain time under optimal conditions by the action of the enzyme. An enzyme that has more activity transforms more substrate molecules into a product in a given time. And the most commonly used enzyme activity unit is international unit, but the system international unit is the catalyst. And we use these both units in the clinical laboratory, but the common one is the unit international, this one. And there are factors, various factors affecting enzyme reactions. The first one is the enzyme concentration. The rate of enzyme reaction under optimal conditions in substrate abundant medium is directly proportional to the enzyme concentration. Another factor, substrate concentration. As the substrate concentration increases, the rate of enzyme reaction also increases. By increasing the substrate rate, a rapid increase in the reaction rate is seen at first. However, as the substrate addition continues, this rapid increase slows down and eventually remains constant at a certain level. Hydrogen ion concentration, the pH. And enzymes show the highest activity at a certain pH, and although this varies for each enzyme, is generally between 5 and 9. And many enzymes in the body has the activity on physiological pH ranges. And to maintain the physiological pH range is so crucial for the enzyme activity, thus metabolism and so on, the life. Temperature. As in other chemical reactions, up to a certain temperature in an enzyme catalysis reaction, an increase in temperature accelerates the enzymatic reaction. Generally, every 10 degrees Celsius increases the enzymatic reaction by 2 times. However, above a certain temperature, such as 45 degrees Celsius, the reaction slows down, they denaturate, they lose the natural structure, will cause the loss of activity. Reaction products. The rate of an enzyme reaction decreases over time due to the increase in reaction products, and this is because the enzyme reaction is reversible. Light and other physical factors. The activity of enzymes can be enhanced or inhibited by the effect of light. Red and blue light increases the activity of salivary amylase and some other enzymes, and UV light, ultraviolet light, has the opposite effect. The time. As the reaction catalyzed by an enzyme proceeds, the rate decreases with time. This is due to factors such as the products combining with each other to form a reaction in the opposite direction, the duration of the enzyme over time, the formation of substances that inhibit the reaction, thus depletion of the substrate. Structure and concentration of ions. The rate of enzymatic reactions is significantly influenced by the structure and concentration of ions present in the medium. This also affects the hydrogen ion concentrations. So there is a strict relationship between the structure and concentration of ions and the pH. Allosteric effect. The end product of many enzyme reactions inhibits the action of the enzyme by a feedback mechanism. Such a form of inhibition is called feedback inhibition, allosteric inhibition, or end product inhibition. And the first enzyme inhibited by the end product is called an allosteric enzyme. Effect of hormones and other biochemical factors. The hormones, amino acids, and other class of substances can affect the state of the enzyme, and this change in the structure of the enzyme can be brought about by various effects, various estrogenic, androgenic, and some steroid hormones. And steroid hormones also have the capacity to modulate the ability of cells to synthesize enzymes. Enzymes are biological catalysts for accelerating a given reaction. And to understand the relationship between reaction rate and the substrate concentration, a model often referred to as Michaelis-Menten kinetics is used. This relationship can be explained as follows. And look at here, we have the graphic showing the relationship between the substrate concentration here at the x-axis, and at the y-axis you see the reaction rate. And as you see, we have a graph here, we have a curve relates with the reaction rate and the substrate concentration. As you see, there is a no direct proportion with the substrate concentration at the reaction rate. What about here at the low level of substrate concentrations? When the substrate concentration is low, most of the enzymes remain idle because not enough substrate molecules are present. And in this case, the reaction rate is directly proportional to the substrate concentration. That is, as the amount of substrate increases, the reaction rate also is increasing. And look at here, we have a high degree of a type of angle of continue to increase the substrate concentration. As the amount of substrate increases, more enzymes interact with the substrate and the reaction rate increases. However, this increase slows down. Look at here, it slows down at some point because most of the enzymes active sites are occupied by substrate molecules and continue to increase the substrate concentration. At some point, all enzyme molecules begin to actively interact with the substrate. And this is the saturation point of the enzymes. And at this point, the reaction rate does not increase further because all the enzyme molecules are already in use. And this maximum rate is called the Vmax. Michaelis-Menten constant, which is the constant m here, point at which the reaction rate of substrate concentration reaches half of the maximum rate. Get here, half of the maximum. The Michaelis-Menten constant gives information about the affinity binding strength of an enzyme to its substrate. A low Km value indicates high affinity and the high Km value indicates low affinity. This relationship has important applications in understanding enzyme activity and in fields such as pharmacology, biotechnology, and biochemistry. In particular, this information is used to understand the effect of drugs on enzymes and to keep enzymes activity under control. Another good illustration showing the enzyme activity at the low and high substrate, or what happens in low and high substrate conditions. And here you see we have 5 moles of enzymes and 2 moles substrate, and increase the substrate as an accepted increase rate of the enzyme activity because more enzymes interact with the more substrate. But at the high substrate level, as you see, all enzymes active sites are occupied with the substrate, thus the rest will not undergo the reaction, and so at that level the reaction rate will be constant, or the reaction rate will not increase. And that's the Vmax. Enzyme inhibitors. Enzymes can be poisoned or therapeutically inhibited by various chemicals. Irreversible inhibitors bind enzymes and, if present, slowly decompose. An example is the nerve agent, the isopropyl phosphofluoridate, which prolongs the action of the neurotransmitter acetylcholine by binding acetylcholine esterase. And reversible inhibitors are usually classified as either competitive or non-competitive. Also, there is an uncompetitive. There are reversible inhibitors and the irreversible inhibitors. Reversible competitive inhibitors. These inhibitors can be thought of as structural analogs of the substrate and therefore compete for the same active binding sites on the enzymes. This is why they are competitive. And because of this competition, the effect of the competitive inhibitor can be overcome if sufficient substrate is provided. Reversible non-competitive inhibitors. These inhibitors are usually not structurally related to the substrate and bind to an allosteric site on the enzyme. This means they bind to a site other than the active site. Thus, both substrate and inhibitor can be theoretically bind to the enzyme simultaneously. However, the inhibitor usually disrupts the active site, thus changing the conformation of its catalytic residue, which further reduces the activity of the enzyme-substrate complex. Since the effect of a non-competitive inhibitor cannot be reversed by increasing substrate concentration, the Michaelis-Menten constant remains unchanged. However, there is a decrease in the apparent Vmax, or the maximum rate. Uncompetitive inhibitors. So be careful. Use interchangeably with the non-competitive and uncompetitive. They are different. Although these inhibitors do not react with the free enzyme, they can combine with the enzyme-substrate complex to slow or prevent product formation. This type of inhibition can be seen in reactions involving two substrates. And here, to make a clear separation between non-competitive and the uncompetitive, let me explain. But the non-competitive refers to molecules that inhibit enzyme activity by binding to a site other than the active site of an enzyme. Uncompetitive inhibitors. This term refers to molecules that can bind to an enzyme and inhibit its activity only when the substrate binds to the enzyme. Irreversible inhibitors. Irreversible inhibitors usually react covalently with an amino acid side chain on the enzyme to form a stable complex permanently inactivate, and some bind to active sites and are known as mechanism-based suicide substrates. And the kinetics of irreversible inhibitors are similar to those of non-competitive inhibitors above. An example is, and also if we use them as a therapeutic purposes, we call them therapeutic inhibitors. And most therapeutic inhibitors are either mechanism-based suicide substrate or competitive inhibitors. And for example, 5-fluorouracil, which is used against cancer, it's a therapeutic suicide inhibitor for the enzyme thymidylate synthase. Enzyme activators. They are also called accelerators. Many enzymes are inactive in cells or secretions. They need to be activated before they can catalyze specific reactions. Some zymogens are activated autocatalytically, or one enzyme acts on the inactive form for another enzyme to activate, and these are most inorganic and sometimes organic cells. And enzymes that require the presence of free sulfide groups in their molecules to function are activated by reducing agents such as potassium cyanide, hydrogen sulfide, glutathione, or cysteine. Coenzymes. There are inorganic cofactors which include various cations such as zinc, magnesium, manganese, iron, copper, potassium, or sodium, and organic cofactors, actually organic coenzymes, which consist of about a dozen substances with different structures. The coenzyme is the active part of the holoenzyme and is directly involved in enzyme catalysis. Many vitamins, especially the B group vitamins or the organic compounds in their structure, act as coenzymes, for example, activators, enzymes that are essentially proteins. Coenzymes are classified according to the groups they transfer. The first class is energy-rich triphosphates, adenosine triphosphate, the famous one, found in all cells, carries energy-rich phosphates, source of energy for many reactions. Active sulfate, PUPS, the biosynthesis of compounds containing sulfate esters, requires activation of inorganic sulfate upstream. Uridine triphosphate, involved in carbohydrate metabolism, formation of epimers of monosaccharides, and also involved in RNA transcription. Cytidine triphosphate, involved in mitochondrial glycerine phosphatide biosynthesis and synthesis of glycoprotein containing neuronamic acid in carbohydrate metabolism. Also involved in RNA synthesis. Guanosine triphosphate acts as a substrate for synthesis of RNA during the transcription process and for the synthesis of DNA during DNA replication. It also acts as an energy source or substrate activator in metabolic reactions. And actually, for the involvement of these energy-rich triphosphates into the DNA and RNA synthesis is common for all. But don't forget, for the DNA also there are deoxyforms of these triphosphates, DATP or DUTP, and it is occurred by reduction of the ribose moiety in these molecules by converting it to the deoxyribose. Group carrying coenzymes, pyridoxal phosphate (PLP). It is the coenzyme of vitamin B6 and acts as a coenzyme in various enzymatic reactions. Don't forget, it is the coenzyme of vitamin B6. And PLP is involved in all transamination reactions and in certain decarboxylation, deamination, and racemization reactions of amino acids. Thiamine pyrophosphate, it is the coenzyme of thiamine or the vitamin B1. It is a coenzyme found in all living systems and catalyzes several biochemical reactions. It is synthesized in the cytosol and is required for the transcatalyzed activity and for pyruvate, oxyglutarate, and branch-chain keto acid dehydrogenase activity in mitochondria. Coenzyme A, it is the coenzyme of pantothenate or the pantothenic acid or the vitamin B5. It is notable for its role in the synthesis and oxidation of fatty acids and the oxidation of pyruvate in the citric acid cycle. Tetrahydrofolic acid or tetrahydrofolate is the coenzyme of the vitamin B9. The vitamin folic acid. It's a coenzyme in many reactions, especially in the synthesis of amino acids and nucleic acid. It acts as a donor of a group with one carbon atom. It's the component of one carbon unit metabolism or one carbon metabolism. Biotin is vitamin B7 and its coenzyme carboxybiotin. Biotin is a coenzyme involved in the synthesis of fatty acid, isoleucine, and valine, and in the structure of carboxylase enzymes and gluconeogenesis. because it transfers or it carries the carbon dioxide, which is why I call carboxybiotin. Cobalamin, the vitamin B12, and there are different coenzyme forms of vitamin B12, such as methylcobalamin, hydroxycobalamin, and adenosylcobalamin. And methylcobalamin is important in the transfer of methyl groups, that's in one carbon metabolism, and adenosylcobalamin is important in the fatty acid metabolism, especially in propionic acid metabolism. Until now, which I talk about the given coenzymes, as you see, they all are the coenzymes of B class of vitamins, but not the lipoic acid. It's another coenzyme, and at least 5 enzyme systems requires the lipoic acid, and 2 of these are in the citric acid cycle. Coenzymes carrying hydrogen, electrons, and oxygen. Coenzymes, they are reducing equivalents. And nicotinamide adenine dinucleotide NAD and nicotinamide adenine dinucleotide phosphate NADP are the 2 coenzymes of vitamin B3. Nicotinamide is the chemical name of vitamin B3. And also we have flavin adenine dinucleotide FAD and flavin mononucleotide FM. These are the coenzymes of vitamin B2, which is riboflavin. And nicotinamide, which is a vitamin B3, is a component of redox coenzymes NAD and NADP. Riboflavin, a component of redox coenzymes FMN and FAD. NAD and NADP, they both act as proton hydrogen acceptors of specific substrates, but they have different fate. NAD mainly coenzyme of the catabolic reactions, while NADP is the coenzyme of anabolic reactions, and they involve in many oxidoreduction reactions. FAD is the enzymes, FAD enzymes, or the enzymes having FAD as cofactor, also involved in the redox reactions, and such as succinic acid to fumaric acid in the TCA cycle. And the ubiquinone, the coenzyme Q, is not a coenzyme or vitamin B derivative. It is synthesized in body. It is found in animals, plants, and microorganisms. It is involved in the electron transport system in oxidation system, or it's the mobile electron carrier in oxidative phosphorylation. In other words, the electron transfer or electron transport system. And we've finished the topic, my dear friends. And here are the references, the books that you can obtain. And with this topic, Biochemistry 1 course, all topics of Biochemistry 1 course have been completed. More about biochemistry, clinical biochemistry, and the laboratory world, you can visit my website, BiyokimyaVET, and also you can follow my Instagram accounts. Please do not forget to subscribe to my channel, to like the video, and to click on the ring bell. Please stay safe and goodbye.